Welcome to this explainer. Today, we're diving into one of the most absolutely fascinating processes in all of biology, how life ensures its own continuation. I mean, from the simplest green algae floating in a pond to the incredibly complex, beautiful flowering plants we see all around us, we're going to unpack the brilliant strategies organisms use to reproduce. Okay, let's get right into it.
Here's our roadmap for today. We'll start with the blueprint of reproduction, look at asexual strategies in nature, explore the flower as a reproductive hub, dive into gamete production factories, and finally trace the journey of pollination.
Section 1. The blueprint of reproduction, cloning versus variation. At its core, reproduction is really just biological recycling, right? It's nature's way of making sure the story keeps going. But there are two very different paths to get there. Asexual reproduction relies on just a single parent producing exact morphological and genetic clones. It's basically a fast, highly efficient cloning machine with no gender involved. Sexual reproduction, on the other hand, requires two parents, specifically male and female gametes, and it introduces genetic variation. You aren't a 100% copy of your parents, and neither are sexually reproducing plants. And that variation, it's the absolute secret sauce for evolution. Section 2. Asexual strategies in nature. How single parents multiply. So before we get into complex flowers, let's look at the incredibly fast-paced strategies of lower organisms. Just look at the diversity here. A filamentous algae like Spirogyra uses fragmentation. If it breaks, every single piece just grows into a new plant. Planaria has this crazy high regeneration power where severed parts grow into completely new bodies. A Hydra? It literally develops a tiny bud right on its body wall that just detaches as a brand new individual. Amoebas use binary fission, where the nucleus divides first, and then the parent cell splits evenly in two. And under really extreme conditions, Plasmodium goes into multiple fission, dividing into tons of cells inside a protective wall, just waiting to burst out. Now, higher plants have their own brilliant asexual tricks, bypassing seeds entirely by using their natural body parts. For example, a potato is actually a stem tuber, an onion is a bulb, and a bryophyllum plant literally grows tiny new plants right off the edges of its leaves. It's wild. And you know what? Humans have actually hacked this system. We use artificial methods like stem cuttings for roses or grafting, where we physically combine the root system of one plant with the shoot of another to mix and match traits like flower color. We even use tissue culture to grow entire plants in a lab from a single cell. And the absolutely crucial concept that makes all those artificial hacks possible is totipotency. It's the incredible ability of a single plant cell to divide and develop into a complete brand new plant. Think about that for a second. Human skin cells definitely can't do this, but a single meristematic plant cell, it contains the complete blueprint and the potential to build a 100% exact copy of the parent plant. It's a real game changer. Section 3. The flower, reproductive hub. Anatomy of the plant factory. We usually just think of flowers as pretty objects, don't we? But biologically speaking, they are highly specialized factories, engineered entirely for sexual reproduction. The outer green sepals protect the bud, while those colorful petals act as giant advertising billboards to attract pollinators. But the real magic happens inside, in the essential worlds. The stamen is the male factory, consisting of a stalk called the filament, holding up the pollen-producing anther. And right dead center is the pistil, or carpel. That's the female factory, safely housing the ovules way down at its base in the ovary. Section 4. Gamete production factories, manufacturing the cells of life. Okay, let's zoom right inside these floral factories for a quick masterclass in cellular engineering. Tracing the male journey, which is called microsporogenesis, we start with a single diploid microspore mother cell inside the anther. It undergoes meiosis to form a tetrad of 4 haploid microspores. Then, one of these microspores divides unevenly via mitosis. This forms a large vegetative cell with a tiny generative cell just floating inside it. Finally, that tiny generative cell divides one last time to form 2 distinct male gametes. And that entire package structure, that is your mature pollen grain. Now, before we move on, we really have to pause and marvel at a substance called sporopollenin. It forms the outer layer, or exine, of the pollen grain. So why does this matter? Because it is quite literally an almost indestructible biological armor. It can't be degraded by any known natural enzyme. This guarantees that these super fragile male gametes can survive incredibly harsh environmental conditions on their perilous journey.
Meanwhile, over in the female factory, inside the ovary's ovule, megasporogenesis is kicking off. It starts similarly, with a mother cell undergoing meiosis to create 4 haploid cells. But here, nature is beautifully, almost shockingly ruthless. 3 of these cells simply degenerate and disappear. All the resources are funneled into just one surviving, functional megaspore. That lone survivor gets the exclusive chance to continue the lineage and build the female gametophyte. And what that lone survivor does next is super fascinating. It's a process called free nuclear division. Basically, the nucleus divides, but the cell itself doesn't split. So one nucleus becomes 2. A second division makes it 4. A third division results in 8 total nuclei sharing one massive single cell. It's only after you have 8 nuclei sharing that space that they finally migrate to specific ends of the cell and build their own distinct cell walls. This whole process creates the mature embryo sac. This leaves us with a final structure you absolutely need to remember: the 7-celled, 8-nucleated embryo sac. At the top, the chalazal end, we have 3 antipodal cells. Down at the bottom, micropilar end, there are 2 synergid cells which will actually guide the pollen tube later, and 1 vital egg cell. That egg is the actual female gamete. And right in the middle, 1 massive central cell holding those 2 remaining polar nuclei. So that's 7 distinct cells, 8 total nuclei, perfectly setting the stage for fertilization. Section 5. The journey of pollination, connecting the factories. So the male gametes are locked in their sporopollenin armor, and the female gamete is waiting in the embryo sac. Let's look at the flight paths pollen takes to bridge that gap. If pollen moves from the anther to the stigma of the exact same flower, we call that autogamy. If it moves to a different flower but on the same plant, that's gyrannogamy. Both of these result in self-fertilization because, well, the genetics are identical. But the real evolutionary goal, that's xenogamy. That's when pollen travels to a completely different plant of the same species, bringing us that crucial genetic variation we talked about earlier through cross-fertilization. Since plants can't exactly get up and walk over to each other, they need couriers, and they rely on completely different delivery agents to get the job done. For abiotic or non-living agents, we've got wind, which scientists call anemophily, and water, known as hydrophily. Then there are biotic or living agents. This includes insects for entomophily, birds for ornithophily, and yes, even nocturnal bats, which is called chiropterophily. What's totally amazing here is how a plant's choice of courier dictates its entire look and design. Wind-pollinated flowers, they basically act like vast, efficient pollen cannons. They're usually small, colorless, or white, with 0 fragrance or nectar. But they produce massive quantities of light, dry pollen, and have these feathery stigmas to catch it as it blows by. Contrast that with biotic flowers. They act as tempting billboards. They have large, brightly colored petals, strong, sweet fragrances, nectar, and sticky pollen that's perfectly engineered to cling onto the body of a visiting bee or bird. So, our pre-fertilization journey is complete. The pollen grain has braved the elements and successfully landed on the correct stigma. But it leaves us with an incredible question. How do those 2 male gametes, locked tightly inside the pollen grain, physically navigate all the way down into that 7-celled ovule to trigger the absolute magic of double fertilization? Well, that is a mind-bending process we are going to uncover in our next explainer. Thanks so much for learning with me today.